Kappa as one of the most economically important seaweeds, is farmed globally for their Carrageenan content. Since 1970s, most cultivars used by seaweed farmers has been vegetatively propagated, and cultivars were sourced from only few strains. This lack in genetic variability may reduce plant vigor, making the cultivars more susceptible to diseases and infection. Furthermore, these farmed cultivars are now observed to have lower productivity, thinner branches, and higher susceptibility to epiphytism and ice disease, thereby compromising Carrageenan yield and quality. In this project, we aim to give solutions to these problems by generating new cultivars from wild population with superior quality. This study will generate new seed stocks obtained from spores of wild Kappa alvarezii, evaluate their growth performance both in land and sea-based facilities, as well as evaluate their Carrageenan yield and quality. The new cultivars with superior quality will then be distributed to seaweed farmers for future cultivation. The parental plants were collected from their natural habitat in Samar, Philippines. The samples were then flown to the Seaweed Culture Lab of the UP Marine Science Institute, Diliman, Quezon City, where they were prepared for spore release experiment. 6 parental plants were chosen to represent 3 carposporic and 3 tetrasporic plants with different color and morphology. Among the progeny of KACRC parental plant, unique morphologies were exhibited. These photos show 9-month-old male and female gametophyte progeny of KATRQ parental plant. Color expressed were dark brown, black, red, bicolor, and orange. The third photo from the left is the 8-month-old presumptive chimeric female gametophyte of KATRQ parental plant. Arrow points to the interface of dark red and dark brown branches. The second objective of our project examines the growth performance of new cultivars. First, the growth and nitrate uptake of 4 different strains of eukymatoids were examined. Euchyma denticulatum and 3 strains of K. alvarezii with different thallus morphologies were used in the study. We found out that growth and nitrate uptake of eukymatoids are strain-specific, and those strains that have high surface area to volume ratio had faster growth and nutrient uptake rates. Further, fast nutrient uptake was translated to faster growth. This is because seaweeds require nutrients in order to sustain normal physiological activities such as growth. Our second study on objective number 2 is about the effects of nutrient fertilization on Kappa alvarezii. Here, we subjected K. alvarezii to pulse fertilization where we have 3 experimental treatments. The control, wherein samples of K. alvarezii were not exposed to high nutrient levels, the 1x treatment, wherein samples were exposed to high nutrient level once, and the 5x treatment, wherein samples were exposed to high nutrient level multiple times. The figure on the left shows that nutrient fertilization enhances the growth rate of K. alvarezii. Similarly, nutrient fertilization enhances branching in the thallus of K. alvarezii as shown in the figure on the right. Our third study under Objective 2 examines the effects of high and low light levels on green and brown color strains of K. alvarezii. Samples of these strains were cultivated at the land-based hatchery of Bolinao Marine Laboratory under high and low light conditions. Our results showed that extreme low light level reduces the growth rate of both the green and brown strains of K. alvarezii. The fourth study under Objective 2 noted the occurrences of epiphytes and endophytes in the land-based culture of Kappa alvarezii. This figure shows the micrographs of the representative epiphytes and endophytes in the cultures. The endophytes infesting our cultures were observed to be filamentous red algae. This endophyte initially formed chains of 3 to 5 cells and is scattered in the cortical layer of its host. As the infection progresses, Chains of these irregularly shaped cells form irregular branches that can penetrate up to the medullary layer of the host Capophycus. The notable results of infection by endophytic red algae is the darkening, roughening, and hardening of host thallus. Endophytic infection also resulted to pearly branch cultivars. Aside from the red endophytes, epiphytic filamentous red algae were predominantly infesting our cultivars. These epiphytes could cover the entirety of the host thalli. But aside from endophytes and epiphytes, in this study we also defined and identified an epiendophyte. Epiendophytes are algal species having initially epiphytic filaments but develop endophytic filaments penetrating into the cortical and subcortical layers of the host at a later stage of infection. Melanothamnus sp is an example of epiendophyte of K. alvarezii. Moreover, weeds and ice disease were also observed in K. alvarezii cultivars. Weeds are algal species that were mistakenly reported as epiphytes but were just entangled in the thalli with no point of attachment. An example of weed is the Chaetomorpha sp. Wounds were observed on the portions infected with endophytes and ice disease. However, these wounds were able to heal and recover when the necrotic tissues were removed. Objective number 3 is about the assessment of Carrageenan yield and quality of Capophycus alvarezii. These figures just tell us that Carrageenan yield and quality did not significantly vary among the 3 nutrient treatments mentioned earlier. It should also be noted that the Carrageenan was extracted using 2 different methods, the native and the alkali or transformed method. Here, the viscosity was measured only on native Carrageenan, while gel strength was measured only on transformed or alkali treatment Carrageenan. The Carrageenan yield and quality of the 2 progenies of gametophytic plant were also evaluated. The 2 strains for GN11 and GN9, wherein no significant difference was observed between the yield, viscosity, and gel strength of the 2 strains. Lastly, objective number 4 is on the distribution of seed stocks to seaweed farmers. To transport the seaweeds, they were first visually inspected. Healthy individuals characterized by the absence of epiphytes and ice disease were chosen for test outplanting. The samples are transported in an insulated cooler, where inside the cooler, cheesecloth were also used in order to maintain the moist environment. This table shows our checklist of the different strains that were sent to Bohol and Iloilo for test outplanting. Raw dried samples of some strains were also sent to Cargill, France for Carrageenan analysis. These are the tetrasporophyte that were sent for field testing, and the table shows their respective growth rates under hatchery conditions. While these are the gametophyte that were sent for field testing with the corresponding growth rates under hatchery conditions. And these are the calculated growth rates in the 2 farm sites in Bohol. What we have presented now is just a snapshot of our entire work. More works are needed in order to generate more resistant cultivars for the farmers without compromising Carrageenan yield and quality. This project has been made possible by our collaboration with the GlobalSeaweedSTAR, Scottish Association for Marine Science, Cargill, and Coast 4C.